DNA replication. DNA replication is the process by which DNA makes a copy of itself during cell division. The first step in DNA replication is to unzip the double helix DNA structure of the DNA molecule. So this is our DNA molecule. We have a 5-prong to 3-prong, and then the tail end of it is 3-prong to 5-prong. So first step is to unzip the DNA molecule with an enzyme called helicase, which breaks the hydrogen bonds. So here is our enzyme, this one right here. It creates a Y shape. It creates a Y shape fork called the replication fork. The two separate strands will act as templates for making new strands of DNA. So here we go. Now we have 2 strands of DNA now. One of them is called the leading strand, which will be this green one. This is our leading strand, and this one is the one that is 3 to 5 directions. The other strand is called the lagging strand, which is the pink one right here, which is a 5 to 3 prime direction. So during the replication process, a short piece of RNA called the primer, which is this guy right here, comes along and bites the end of the leading strand. The primer acts as a starting point for DNA synthesis. DNA polymerase binds the leading strand and then walks along it. So right here, this is going to be your DNA polymerase. So our primer is right here, and our primer is going, and our DNA polymerase, it's continuing to take this leading strand over and binding with it like little teeth. This sort of replication is continuous because it just continues on and on and on and on and on. It's straight. It's our leading strand. The lagging strand, however, it's a different story, which is this guy right here. Our lagging strand, it's not done in a continuous manner. Numerous of RNA primers are made. So our RNA primer, we're back in our fork. Our RNA primer is made and and binds at various points along the lagging strand. Chunks of DNA called Okazaki fragments. So these guys right here are going to be our Okazaki fragments. What? So our Okazaki fragments, chunks of DNA are called Okazaki fragments. They are then added to the lagging strand, also in a 5 to 3 direction. So our Okazaki fragment will. So numerous RNA primers are made by the primase enzyme and bind at various points along the lagging strand. So it binds here, it binds here, and it binds here. Chunks of DNA called Okazaki fragments, which will be these fragments right here, are then added to the lagging strand, also in a 5 to 3 direction. This type of replication is called discontinuous, as the Okazaki fragments will need to be joined up later. Once all the bases are matched up and the enzyme called exonuclease strips away the primers, so then these primers all get stripped away. This way, sorry. Get stripped away. The gaps where the primers were are then filled yet by another complementary nucleotides. The new strand is proofread to make sure that there are no mistakes in the DNA sequence. Finally, an enzyme called DNA ligase seals up the sequence. So all of these guys are finally spread into 2 strands. So the DNA ligase seals up the sequence of DNA in 2 continuous double strands. The result of DNA replication, the result of DNA replication is two DNA molecules. Two DNA molecules, which are done in a semi-conservative process. Half of the chain is part of the original DNA, which is right here, pink chain, and the yellow one, it would be our new DNA molecule. It's in here. Half of our DNA molecule will be this green one, which is our original, and this darker yellow, it's our new one.